Okay. Good afternoon. I am Supriya Naika, MK, your host for this captivating program brought to you by Creactive. In our 120 episode journey, Creactive stands as a community of like minded individuals driven by curiosity and a passion for acquiring new knowledge. The essence of Creactive lies in its name, a fusion of creativity and activity. We believe that to be truly active, we must embrace creativity. At the heart of our mission is the knowledge square, symbolizing the belief that sharing knowledge enhances our collective wisdom. Creative vision is to spread constructive thinking across various domains with a particularly focus on non-textual, non-academic and non-syllabus concepts. Every Saturday we gathered for our online weekend talks on the Zoom platform, broadcasting live on our YouTube channel Creactive GPD. We extend our sincere gratitude to our resource person for generously sharing their knowledge, and we invite you all to active participate by asking questions and engaging in discussion at the end of each session. Today we are honored to have Dr. Vignesha Radha with us, a distinguished scientist at CCMB. Her extensive research has significantly advanced our understanding of cellular functioning, particularly in regulating cell fate at the molecular level. With a post-graduation degree in human genetics from Andhra University and a PhD from Institute of Genetics in Hyderabad, she has made significant contributions to our understanding of molecular level cell fate regulation. Her research spotlight on how cells specialize, undergo programmed death, and respond to signals, crucial insights into disease mechanism. Having mentored numerous PhD students and authored over 70 papers, Dr. Radha ma'am is not only a distinguished scientist but also passionate about science popularization. Our title today is Cancer, Why and How, a topic of immense importance. Dr. Radha ma'am will unravel the complexities under the control of various factors and signalized pathways, explaining the decision cells make, whether to profile rate, differentiate, or face programmed cell death. Join us in understanding the molecular intrinsicy and novel approach in cancer treatment. Not further delay, we welcome you, ma'am, for today's session, and I welcome all the participants of Zoom and YouTube viewers. We welcome you all, and over to you, ma'am.
Thank you, Supriya. At the outset, let me make a disclaimer. When you look at this title, please don't get carried away thinking that I will provide answers to every question you have about why and how of cancer. We are still in the middle of a long, long journey to understand cancer because it is not a single disease. It's a heterogeneous group of disorders that have all been clubbed and given the name of cancer. You must have come across somebody or the other who has recently been diagnosed with cancer. And the first question that crosses their mind is, why me? Why did I get this disease? That is what cell biologists have been trying to explain, and their attempts have been partly fruitful. And if I have to give a one-line answer as to why me, it's because there have been some genetic changes in the cells in this individual's body. that have lost normal regulation. Okay, let me through this talk tell you some fundamentals of how complex multicellular organisms are built, because in multicellular organisms, every cell has to be programmed to perform a specific function so that it contributes to the whole of the body, where there is distribution of tasks. Your liver performs one kind of task, your heart performs one kind of task. So you are all aware that all organisms have started with a single cell. Now, this single cell in a body like ours is responsible for forming the variety of cells with very, very specialized functions. A brain cell is so different from a blood cell, which is again very different from a bone cell. Now, how does this happen? If we understand this, we understand the mechanisms of what goes wrong when we have cancer. The simple answer to how from one cell a body is formed is that our cells divide. Yes, all of us are aware of that, because in an adult there are about 10 to the power of 13 cells where we started with one cell. Now, how you think that if the single cell is dividing, that simple textbook knowledge is that a mother cell divides into two daughter cells. Now, if that was happening, at the end, you would just have a bag of equal or similar looking cells. That's not what a body looks like. The body has a variety of cell types which are grouped together as different types of tissues with very, very specialized functions. Now, how is it that a single cell forms so many different cell types? The reason is that there is a programming which decides the fate of every single cell. And where does this programming come from? Just the environment of the cell and also the genes within the cell. So when I say environment, in response to the extracellular environment, it might be a neighboring cell, it might be a chemical signal, it might be a hormone. Okay, so these signals are received by the cells, and then that results in changes in gene expression, which tells the cell whether it has to divide, differentiate, stay quiet without doing anything, without dividing or differentiating. Differentiating means attaining specialization, simply put, okay? And then also whether it should undergo cell death. The scientific term we use for cellular death is called apoptosis. So which is different from death due to injury, because here it is a programmed cell death where the cell is voluntarily dying in the benefit of the organism. Okay, so these changes in the cells eventually result in formation of tissues and the body as a whole. So several years, several decades actually, it has been a curiosity factor for many cell biologists how this happens. And this has been my line of work over the last several decades. So let me begin with something very simple. I'll show you how a cell receives a signal. Everything is primarily dependent on a variety of proteins. The signal is also usually a protein. It can be a small hormone circulating in the blood, or it can be another large protein sitting on an adjacent cell. Anything can be a signal. It can also be a mechanical force. In some cases, it can be physical signals like light. Okay, how we perceive light in the eye, these are also signals. So when the cell receives a signal, there is a receptor which detects the signal, and passes on the message to a series of proteins, which eventually tell the functions in the nucleus, what they have to do, whether they have to make a new protein, and whether they have to secrete a molecule, whether it has to undergo shape changes, okay, and attain specialized functions. And not the least, whether it has to die also in response to some untoward signals. The cell has to die in the interest of the tissue and the body as a whole. So, as I mentioned, before we get on to this disease called cancer, it is important that we understand how cells divide. Very, very simply put, cells divide in a unidirectional fashion by undergoing changes. What are these changes? What are the most important changes? The size has to increase, the cell has to grow, the DNA has to duplicate. Okay, you can't have the DNA making some parts of the DNA having more copies, some less copies. Every single base pair has to duplicate, not one more, not one less. Okay, and all the 6 billion bases. And then what should the cell do once the DNA has been duplicated? It has to distribute the duplicated DNA into the daughter cells. And that's what is shown here as a cycle. So we call this the cell cycle. So there are molecular controls which determine this, how the cell should grow, how the DNA should duplicate, and then how the duplicated DNA should split into 2 cells. Sometimes when the cell doesn't want, where the tissue says stop, we shouldn't be dividing anymore, the cell goes into a resting state called we call it the G0. So there it is neither dividing, but if necessary, in response to signals, it can come back in and reproliferate, enter the cell cycle again. Now, in addition to this kind of control that enables the cell to divide, there is another kind of control which is a monitoring kind of control. At each step, it will monitor, has the cell grown enough in size? Has the DNA duplicated? Is it intact? Are there any defects in the DNA? Has all the DNA replicated? Is the DNA within one chromosome not replicated? And then, because for division, equal division of the chromosomes into the daughter cells, they have to align properly. So they come together and then move into the 2 daughter cells. So this kind of monitoring checkpoints are there. And what do these do? If they find any defect, if they find that some of the DNA is not replicated or there is a damage to the DNA, it will trigger a stop signal. And that happens at multiple places in this cell cycle. See, the actual division into 2 daughter cells is a very small time frame of the total cycle of cell division. The cell spends a lot of time in the growth phase where it is duplicating all its resources. All the proteins in the cell, all the lipids in the cell have to multiply because they have to go into 2 daughter cells. So it spends a lot of time in this growth phase. Then it spends some amount of time when the DNA is replicating. And then there is a little gap which makes sure that all the DNA is replicated. So you need these phases to monitor whether this event, the primary event is division of DNA and then segregation of the chromosomes. Okay, but then these phases are important. Now, if the chromosomes are not aligned properly, then you have a stop signal which says that the daughter cells should not divide. So these kind of monitoring systems are called checkpoints. Why am I mentioning this? These are very important to ensure that the cell is dividing at an appropriate time and stopping when it has to stop dividing. So if there is a defect, what will happen if a cell that has defects in its DNA continues to form daughter cells? That defect gets propagated. Okay, and we know the consequence of defects in DNA. So almost like if you consider the scale of operations, there are 6 billion bases in each of our cells. And I mentioned that on average, we have 10 to the power of 13 cells. In each of our cells, whenever it undergoes division, these 6 billion bases have to replicate correctly. Okay. If you didn't have a monitoring system, can you imagine there is an enzyme that actually is responsible for replication of the DNA? And it is quite natural that there will be errors. This monitoring system is in place to detect these errors and tell the cells to stop so that there is time for either repair. Okay. Now, I will just give you an example of how do cells know when to stop. Okay, there are multiple signals. I told you there can be a signal from a neighboring cell. Many of you would have got, small injury or cut in your hand. So what happens when you have a cut in your skin? What you see is that the cut has healed and everything is fine within a few days. How does that happen? What has happened? The cut has triggered the cells which have been staying quiet to start dividing and grow again to patch that gap. But does that mean that they are continuously growing? What tells them to stop when they touch each other? They stop. Otherwise, you know what happens if they continue to grow when they touch each other? What will happen? There will be three-dimensional growth. And I'm sure you would have seen people with some defects in their skin called keloids, which are small, tiny growths on their skins. Okay, so that doesn't happen. Most of the time, most of us don't have this kind of abnormal growth conditions. So how does that happen? When there is a cut, the cells in the border of that cut are triggered to grow. And then when they come together, there is a stop signal which tells the cell, now you don't proliferate or don't divide anymore. So that is how cell proliferation is kept under check. This is one example where the cells are stopping in response to and the presence of an adjoining cell. That is called, in scientific parlance, we call it contact inhibition. All normal cells in our body are programmed to stop their cell cycle when they reach in contact with other cells. Okay. Now, basically, as I mentioned, by and large, our cells, most of our cells are in a differentiated state. That means they have attained specialized functions and they're carrying out their functions. The bone cell, a cartilage cell, a nerve cell, a muscle cell in your thigh, a skin cell, a liver cell, a heart cell, they're all highly specialized and by and large, they're not undergoing division. So, nature has programmed in such a way that the cells in our body are highly resistant to abnormal growth because we have something which I described as a checkpoint control. Okay. So now what happens if this regulatory mechanism is not there? Then that's what you will have. You will have excess of proliferation, sometimes less of proliferation. And if cells that have been told to stop are not stopping and continuing to divide, then that is again leading to excess proliferation. That is when you have abnormal growth, which can be cancerous. not all abnormal growth need be defined as a cancer. I will come to that in the next part of my talk. Okay. So basically, what does it mean? Cancer cells do not listen to stop signals. And they have also acquired the ability to continue to grow even when there is no signal telling them to grow. Because there have been, as I mentioned, there have been some changes in their genetic makeup that is telling them that your control is lost, you continue to grow. So basically, they have pressed the accelerator, but they don't have a brake. Okay, now I'll just show you how a normal cell looks and how a cancerous cell looks. A normal cell, as I mentioned, here there are two types of pictures shown. So when you grow normal cells, when they come close to each other, they will stop dividing. Only the ones in the periphery will be dividing, where there is space for them to grow. But a cancer cell, you'll see, it is growing in 3 dimensions, one on top of the other. Okay. And another thing is they can just change shape and start acquiring new functions. New functions means they can move easily from one place to another. That is because these cells are not undergoing cell death or stopping to divide when they have been told to divide. So this deregulated cell death is very often the cause of cancer. That cell has a genetic abnormality. Normally, such a cell should get eliminated from the tissue to save the tissue and the body. But its dying mechanism is not getting operational. So it continues to divide with its defects, and then you will have not only mutational changes. Mutation means, I mean, single changes in the base pairs of the DNA, but you can also have chromosomal abnormalities. You can have shorter chromosomes, you can have longer chromosomes, you can have duplicated chromosomes. Okay, so which is called polyploidy, and that is a very common feature of cancer. Okay, so you can define cancer cells as abnormal cells that had developed resistance to many of the signals governing cell growth and death through the accumulation of genetic mutations. This is basically how we define cancer scientifically. Now, I told you at the beginning that I will not be able to give you answers to how and why of all cancers. Why is that so? Because there are multiple, nearly 200 types of cancers that have been identified, because they can arise in any tissue. And the cancer in any different tissue is different. And in the same tissue of different individuals is also different, because the genetic changes in each of the cells can be different. When I say cells, these are basically the changes have happened in the cells of our body, not in our germ cells. Germ cells means reproductive cells. Okay. So because of the complications involved, the multiple genetic changes involved, cancer at one time was thought intractable. We cannot do anything. But in the last 50 years, multiple developments that took place, we have been able to culture living cells outside our body. Then there has been many molecular tools have become available, and there's been more crosstalk between clinicians and scientists, and we have been able to understand what exactly is happening when an individual has a cancer. Not a full understanding, but to some extent. So I'll just give you a brief history. If you are misguided in thinking that cancer is only seen nowadays, only in the present generation, or only in the last 50 years, you may be wrong. Cancers have been detected even in mummies and in fossils of humans several thousands of years old. And the word cancer itself was coined by Hippocrates, meaning a crab in the Greek language, because I think he noticed when he did an autopsy, he saw the tissue had lots of blood vessels which were, percolating the tissue. Because for cancer to grow, you need heavy supply of nutrients. So every cancer tissue that is growing has extra blood vessels. So that's how he named this kind of disorder as cancer. And it is ubiquitous. If you think that cancer happens only in humans, no. Many other organisms, whether it's dogs or cattle or wildlife, can also have cancers. Basically, because it is a disorder of an abnormal growth of cells due to loss of regulation. So multicellular organisms, that is primarily metazoans, can develop cancers. And presently, the incidence is quite high, and it has become, cancer has been the cause of 1 in 6 deaths globally. And primarily, we can call cancer as a genetic disease because it is caused by changes in the genes. That doesn't mean, when I say genetic disease, that doesn't mean if I have cancer, my children will get cancer. Okay, please don't think that it is inherited. There is a small component that is inherited, but by and large, cancer is not inherited. Okay, but it is because of changes in the genes which have taken place in the cells of our body, whether it is in the eye, whether it's in our oral cavity, or in our bone, or in our blood. So these cells don't get transmitted to the next generation. So for that reason, it is not necessarily, only a small proportion of cancers or a small proportion of changes in genes are inherited. Okay, and based on the tissue type where the cancer has formed, where it has originated, cancers can be classified. You must have heard breast carcinoma, colon carcinoma, cervical carcinoma, or you must have heard leukemia. These are some of the common, so based on which tissue. So leukemia means it is a cancer of the white blood cells which have propagated unduly and have spread, and that causes a lot of damage. You can have anemia and then multiple other defects. Similarly, sarcoma, connective tissue, that means tissues like the bone come under connective tissue. They are called, when the cancer originated in this tissue, it is called a sarcoma. So some of the properties of cancer cells, it is important to know. How are they different from normal cells? So one by one, I will just list them. These are the primarily important features of a cancer cell. And that is, it sustains a proliferative signal. That means continuously keeps dividing. Then it evades growth suppressors. It avoids, see, normally if there is an abnormality in the body, our immune system doesn't like it. It tries to fight it out. So cancer cells are not like normal cells. They express different kinds of protein in their cell membrane. So our immune system normally will fight it. But what have cancer cells done? They have made a plan to make sure that the immune system doesn't attack them. Okay, so that is what is called by evasion of the immune system. And then normally cells will not grow perpetually. There is a limit on the number of times a normal cell can divide. But cancer cells are not like that. A change has taken place which enables them to become immortal in that sense. Okay, that means they can undergo any number of divisions. Then the very important feature of cancer cells, what we actually call cancer or malignancy, is that they can invade and move from one place to another. That means they can go into other tissues and establish themselves there. Normal cells, a liver cell, if you take it out and put it near a bone, it will not grow. But cancer cells have this ability of growing in multiple niches. That means wherever they are, they can grow. They don't need a suitable environment. They have been programmed internally to continuously grow wherever they are sitting. That's why you have metastasis. And changes have taken place in the shape of these cells that enables them to move out of their tissue into the blood vessels, flow through the blood, and again come out of the blood vessel and establish themselves in another part of the body. That's why somebody with a breast cancer can also develop a brain tumor. It can metastasize to the lungs or the brain. Okay. Then the other property that is very important for its survival is angiogenesis. That means creating a lot of blood supply, vascularization. Okay. And then very, very importantly, it resists cell death. Okay. And cancer cells, the metabolism of cancer cells is changed so that it doesn't depend on energy supply, that is ATP, like normal cells. I won't get into the details of that, but take it from me. These are the primary changes. So you can have changes when the change is in a protein or a gene that enables proliferation. That gene is called an oncogene. When the change is in a gene that is important for suppressing cell division, then it is called an anti-oncogene. Eventually, cancer will show up only when these anti-oncogenes or these checkpoints or these monitoring systems are non-functional. So generally, if a person has cancer, there have been multiple changes, genetic changes that have happened. There is a triggering of proliferation and also a suppression or a defect in its monitoring system. Okay. That is why treating cancers is also difficult, because the cancers have undergone a multitude of changes. Typically, what happens? There can be a genetically mutated cell. So where there is an oncogene that will start proliferating to some extent. But now, normally these cells should die out because they should not undergo excess proliferation. But again, there is a change, another mutation in or dysfunction of a monitoring gene. Then what happens when the monitoring gene is not there? These cells are very happy. They can continue to grow. And that's when you have a tumor growth. And then another kind of change happens that enables them to move from one place to another. That kind of change happens generally in the cytoskeleton, that is the skeleton of the cells. There are proteins which maintain our cell shape. Okay, because a nerve cell is very long. There is something like we have a skeleton in our body. The cell also has proteins which give firmness to the cell, which enables the cell to move from one place to the other. So that's the final change, and that's when you have metastasis, and that's when we define it as malignant. Okay, when it causes a real damage. Sometimes there can be a benign growth which is just stuck there and is unable to move. Then it is called benign. It is not usually called a cancer. Okay. So I have just mentioned some of the differences with properties of cancer cells. And primarily these are the properties by which normal cells and cancer cells differ. See, because they can grow in the absence of signals. They can ignore signals that tell them to stop. They can invade nearby areas. So they can vascularize by telling blood vessels to grow towards the tumors. They can hide from the immune system. And they also undergo chromosomal aberrations because the monitoring systems are defective. Okay. So I should just briefly mention, since we have mentioned the properties of cancer cells, how do we study them in the lab? As a scientist, I think I wish to, at least briefly tell you, how can we study? How can we define if we have a cell growing in the lab? How can we say it's a normal cell or a cancerous cell? Okay, I told you a normal cell, as it grows, when it comes in contact with another cell, it inhibits, it doesn't grow further. It will just form a layer and stop. Okay, but a cancerous cell will start growing now in 3 dimensions. Okay, that is one way. So it is actually defective in contact inhibition. And then it can also grow in 3 dimensions. Okay, and the other thing is normal cells, we have to add a lot of proteins called growth factors. Only then the normal cells you can keep in culture. But cancer cell, even in the absence of growth factors, it can continue to grow because internally in the signaling molecules, one of the signals is permanently on. It is not getting on only in response to a signal. So even in the absence of signal, this is on. So it continuously tells the cell, go on dividing, go on dividing. Okay. And another way of studying whether a cell is cancerous is to introduce into an animal model. Usually we use mice and see whether it can grow there inside the tissue and also whether it can metastasize. That means it can move from one tissue into the another location. Okay. So this is generally how we study cancer cells. So various properties I mentioned, all these properties are dependent on signaling. Signaling means how information is transmitted from the outside of the cell to the inside of the cell and how the cell is responding accordingly. So mostly defects are in the signaling molecules and in the monitoring molecules. So even in response to the signal is usually to tell the cell to divide. Okay, either you can have defects in these signals or you can have defects in the machinery of the cell cycle. Machinery of the cell cycle has the basic machinery plus the monitoring machinery. Okay, so many of these the cancers are because of defects in, and while I said, so here I have indicated them as blue and red. The blue ones are the ones that are oncogenic and the red ones are the ones that are anti-oncogenic. Okay, so when we use drugs, that what you want to do, you want to repress the oncogene and you want to upregulate or activate the anti-oncogene. So here is an example. I said most of our cells are in a differentiated stage, whether it's your skin or your bone. Now, if there is a defect, that the cell is not staying in a specialized state, it does not want to specialize, it just wants to grow without control. See, some of the cells at the base of your skin are actually enabling growth. As the layers build up, it differentiates to form the skin. Now, if this differentiation does not take place, differentiation means forming, in the case of the skin, it is to form a layer and attain all the hair cells, the sweat glands, and things like that. Okay. Now, if that differentiation is not taking place, you can have a variety of disorders and cancer primarily because the cells in differentiation means the cell is stopping to divide. Only when it stops dividing, it can differentiate. So these are just some of the examples. Many skin disorders, including psoriasis and keratosis, are because of defective differentiation. Okay. Now, I want to emphasize a little bit on this checkpoint or this controlling monitoring system. And there is one protein within all of us which plays a very, very important role in monitoring. And that protein is called P53. Okay. This protein's function, it is not normally particularly present in the cell. It is made, but it is removed also. It keeps on getting recycled. Okay. Now, what is the function of this protein? It starts functioning when it sees any kind of stress or damage. The stress can be like, it can be nutrient deficiency, it can be hypoxia, it can be DNA damage, it can be overproliferation of cells, it can be oxidative stress. Okay, so any kind of stress ensures that P53 suddenly gets activated. What is the function of P53? Its function is to arrest the cell cycle. How does it do it? It enters the nucleus and triggers synthesis of all the proteins required for arresting the cell proliferation. First thing P53 will do when it's activated is stop. Don't proliferate at all. Now, once it is stopped, there are multiple options for this cell. One option is if the damage is small, the cell does not want to waste itself. So it just repairs. So P53 makes sure that the repair proteins are readily available and they repair the damage. But if the damage is such that it cannot repair, then P53 makes the molecules required for that cell to die. So so that a defective cell does not proliferate. See, you have billions of cells. Even if one or two cells are lost, it doesn't really matter to the tissue. But if that cell starts propagating, then it becomes hazardous. So this is a safeguarding protein which enables us to stay very, very healthy.
We wait for a while. Maybe some network issue. I'll come back. Stay tuned. Ma'am is coming back. Some network issue. We have class till. If any questions are there, please write down in chat box, or you can ask directly at the end of the session. And I think ma'am is coming back. Every weekend we organize online talks. If you want to join our group, I'll post our WhatsApp group link and YouTube channel link in chat box, so that you can be in contact with our regular activities. Check your chat box. Live streaming link is available. Reactive WhatsApp group also link is posted. Okay, 1 minute. Yes, ma'am. I made you co-host. Radha, ma'am.
Yeah.
Yes.
Yeah.
Yes.
I'm sharing. Yeah.
Mm.
Can you see it?
No, ma'am. Yes, ma'am.
Okay. Yeah.
Yes, ma'am. Okay.
Sorry, everybody. You can hear me?
Yes, ma'am. Yes, ma'am.
Okay. Yeah. Okay. So I was mentioning about the properties of P53. It is an extremely important guardian protein, and very often, because it plays a role in almost any kind of stress response, most cancers in humans are due to defective P53 at the end stage. It can be a defect in the P53 itself, or anything that is regulatory of P53 downstream. So P53 signaling and regulatory proteins, if they are defective only, you will get cancer. So 90% of all human cancers are because P53 is unable to signal, either because there is a mutation in it or the downstream molecules are defective. So when P53 is mutated, it causes drug resistance, it avoids cell death, it causes genomic instability, and all the other properties required for human cancers. So that is why I wanted to stress, and that is one of the reasons it is P53 is called guardian of the genome. Okay. Now, we have literally over the last 3 or 4 decades struggled to understand these mechanisms that I've just been talking to you about. How the cells proliferate, what molecules tell the cell to proliferate, what molecules are checkpoint control regulators. Okay. But for millions of years, viruses have been living with us. Okay. And they have deciphered which molecules in our cells have to be controlled if they want to invade and cause cancers. All of you are aware that some cancers are caused by viruses. Okay, how do these viruses know which molecules to affect, to deregulate? So what does the virus do usually? It makes a protein that tells the cell continuously divide. Okay. For example, if you take the human papilloma virus that causes cervical cancer in women. Okay. So it makes a protein called E7, which increases proliferation. But does our human body like that? When it sees that some cells are over proliferating, it kicks up our guardian protein. What is our guardian protein? P53. What does P53 will ensure that that cell is eliminated. When a cell is infected in our body, the body does not like to keep it. Its first response is to eliminate that by cell death. But what does this virus do? It wants to invade our cells. Okay. So what does it do? It makes another protein called E6. Now, what does this protein do? It can actually latch on to P53 and prevent it from functioning. So if P53 is not functioning, what happens to those cells? They will continuously keep proliferating. So there are many viruses which invade our body. Some of them cause cancers. Okay. And by and large, all of them act by mimicking molecules that enhance cell division and inhibit cell death. Okay. And generally, cancers that are caused by viruses depends on the load of the virus, how long one has been exposed to the virus, etc. Okay, I've just shown in this table the various types of cancers that can be caused by viruses. Some of the common cancer-causing viruses are the papilloma virus, the hepatitis C virus, the Epstein-Barr virus, etc. Okay. So now we have achieved some bit of understanding of why these cells are totally deregulated and continue to divide. I think now to some extent, you know the answer. It is because some of the proteins that are telling the cell to proliferate are overdoing their job, and the ones which are telling the cell to stop are non-functional. Earlier, we used to treat cancers just by inhibiting the growth of all proliferating cells. Because even though our body has some essential proliferating cells, to treat the cancer, it was important to blanketly block all the proliferating cells so that the cancer gets killed. That is what chemotherapeutic drugs that were used earlier were doing. So there were 3 options. One option was surgery. If you have a solid cancer, you can remove that and remove a little bit of the surrounding tissue. The other one was to kill the cells either by radiation or by chemical agents. So radiation is still local, but just to rule out that this cancer has not gone through the blood and stuck here and there, the advice is to undergo chemotherapy. You must have come across somebody or the other among people known to you have undergone chemotherapy. Earlier chemotherapy meant blanket killing of all the cells. So that is why the cells, good cells that are required to proliferate. For example, our intestines have cells that proliferate. Our hair cells have to proliferate. There are some cells which have to continuously keep proliferating. Now, all those cells also die. That is one of the reasons why chemotherapy induces hair loss. It induces difficulties in the digestive system because the proliferating intestinal cells are all killed and absorption of nutrients is very, very poor. Okay. But because now we have identified some of the molecules and we understand some of the mechanisms of how cancer cells have attained specialized properties, we have new therapeutic methods. So suppose you know there is a form of leukemia which is due to overactivity of an enzyme called ABL. That is called chronic myeloid leukemia. So this enzyme, because it is an enzyme that enables cells to proliferate unduly. So our blood cells, white blood cells will continuously proliferate. Because we know that this enzyme is responsible, people have designed a molecule that can sit and block the activity of this enzyme. That has achieved a lot of success for leukemia patients. Okay, this is just an example of how, if you know which molecule is primarily responsible, these molecules are called oncogene addiction. That means this molecule, if you remove this molecule, the cancer is addicted to this molecule. If this molecule is non-functional, the cancer will not survive. Okay, so same way with P53. When P53 is mutated, now we have mechanisms that can, to some extent, correct some of the changes that are there in P53. Okay, so these are the new modalities of treatment. So while chemotherapy is toxic to the cell and kills the cell, you can have hormone therapies which are actually cytostatic. That means they just simply arrest the division of the cells. Okay. Now, other treatment modalities are, because the cancer cells are evading the immune system, we are trying to do immunotherapy to make sure that the immune cells are programmed to detect the cancer cell and kill it. These are called, these come under gene therapy modalities. Okay. So I have listed here some of the treatment modalities which will act on each of the. So what kind of inhibitors you can use to suppress growth? What kind you can use to inhibit metastasis? Similarly, what kind you can use to inhibit angiogenesis? Now you have a variety of drug types. These by and large are all fall under enzyme inhibitors, cell death inducers, okay, and angiogenesis inhibitors. So these are basically called precision therapies based on which molecule. These are multiple signaling pathways I am showing that are generally defective in cancerous cells. So, and to each of them, we have now are having specialized inhibitors that can block their function. So these are called precision therapy. But this, it works very well. It prolongs survival, but it is causing heavy burden on the patients because the treatment is extremely expensive. Okay. And also, because of multiple changes, only the primary tumor is easily targeted. If this tumor has acquired various other properties and moved and located in some other place in your body, then the treatment is again difficult. You should have some other therapy for that. So at the end of the day, single therapies are not functioning for cancer. You should have combination therapies. And another problem that we have come up with this precision therapy is, I just mentioned this enzyme called Abelson that is responsible for chronic myeloid leukemia. After some time, this cancer cell, what does it do? There is a mutation near the enzymatic site, which does not enable the drug to block the activity of this enzyme. So it can escape. So basically, it is refractory to treatment after some point of time. So first 10 years, people were very happy with this inhibitor. Chronic myeloid leukemia can be treated. But after maybe 5 years or so of treatment, new mutations come up in this gene, in this enzyme, which don't allow the drug to act. It is just like you have this microbial resistance, antimicrobial resistance. Similarly, resistance to chemotherapeutic agents. That is also happening in the case of other examples like P53. So I mentioned the two novel immune response kind of treatments, immunotherapies, are one is to target the surface of the cancer cell, which has special properties to evade the immune system. So there we are actually bringing in inhibitors that will enable the cell to be recognized. The other kind is something which is partly a cell-based therapy, where you can make chimeric molecules of the cancer protein, cancer cell protein, unique specialized cancer cell protein, and fuse it and introduce those cells again into the body, so that those cells will now, that immune cells will now, these are T cells in our immune system, cytotoxic T cells they are called, they will now attack only the cancer. So the advantages of these new methods is that the normal cells are not disturbed. So the side effects of these therapies are very low because only the cancer cells are targeted. Let me also bring up another point. Viruses have the property of lysing cells. They enter the cell, multiply, and lyse the cell. So this same strategy is now being, this is one of the new methods, is now being used for treatment of cancer. And these viruses are programmed to attack only the cancer cell and not a normal cell. Now, how have we come up with these new therapeutic modalities? We use the cancer tissue itself. We study its properties, we take out this tissue, find out what is different, sometimes grow them in the lab, try all the drugs on this lab-grown cancer, and then take it back to the patient. Sometimes, you know, a patient is refractory to ABCD drugs, which are generally given. Then what do? Then you test other novel therapies, other immunotherapies, other modalities, and then that has to be tested in the lab. You can't give it to the patient directly. Okay. So the cancer tissue has been helping us in understanding how best to treat a particular cancer. Now, there are many hurdles. See, usually some of the cancers, the patient presents at a very late stage. Then sometimes the diagnosis is difficult because we don't have very good methods of diagnosis for some kinds of cancers, or early diagnosis is not there. And then how do we handle metastasis? Overall, the socioeconomic burden of cancer is very, very high in our population. And in a country like India, where the population is very high and the costs are not affordable, the load is even more. Okay. So the approach now, parallel approach now, is to try prevention. So you can prevent only if you know what to do to evade the having cancer at all. So there are only some environmental factors which we have proven evidence of causing cancer. Tobacco smoke is one, and then also chewing tobacco is the other. Okay. And the other way, so you can avoid, if you identify an object, you can avoid. Then the other thing is, like, you know, go in for secondary mass prevention by mass screening. Like that's what they've been doing for cervical cancer and also for breast cancer. You can go for Pap smear in the case of cervical cancer and give a vaccine, because cervical cancer is because of a human papilloma virus. So now we have a vaccine, and that has proven very, very effective. And there are some dietary associations. Like, for example, if you high sugar, salt, animal fat seem to be associated with higher risk. And a diet with high fiber and having antioxidants seem to be associated with a lower risk of cancer. But these are all dependent on different population studies. And it is not anything that, okay, I will not take sugar at all, I will not get cancer. That's not the way one has to look at it. These are just associations. Okay, so very often you have this is can be associated with many, many conditions. So it need to be cancer. One doesn't need to worry that because of cancer. Okay. If your immunity is low, there's now evidence that cancer survival of the cancer tissue is okay. So the prognosis is more difficult. So there is an immune relation with cancer. If you are totally sleep deprived and you have other ailments, then there is a possibility that the cancer that is triggered in your body can grow and become malignant. Now, is stress associated with cancer? This is a very difficult question to answer because there are no foolproof ways of examining that. But one indirect connection is that stress is associated with lower immune function. And because lowering immune function can aid cancer, we can indirectly say that there is a component or a relationship between stress and cancer. Now, longevity and cancer. What do I mean by longevity? Animals that live longer, do they develop cancer more often? To some extent, we thought it was true, but not necessarily so. Because, but is the body size associated with cancer? Because bigger animal body size doesn't mean bigger cell size. It just means more cell number. More cell number means the number of cells in an elephant have all arisen from the elephant's egg, single cell. But the elephant has many more number of cells than a human body has. Does that mean, what does it mean? That egg cell has undergone that many more cell divisions at the end of the day. But there are some reasons why you would assume that an elephant, because it is undergoing more cell divisions, more defects in the DNA, so likelihood of having cancer is higher. Yes, true to some extent, but we have recently discovered that the elephant has many, many copies. We have only one copy of this P53 protective gene, whereas elephant has nearly 20 copies of this P53, the guardian gene. So it is protected much better than us from cancer. These are some general queries, but I thought I will just mention some myths or some wrong misconceptions about cancer that causes needless worry and hinders, management of that individual and treatment decisions. See, cancer is not a death sentence, certainly. A 5-year survival rate for some cancers like breast and prostate thyroid cancers is nearly 90% or even better. And 5-year survival rate of many other cancers is quite high. It's nearly 70%. Only few cancers are very, very difficult to manage. And one of them is pancreatic cancer. But the new modalities have at least helped to prolong the survival after the detection of the cancer. And just like I said, there are no studies which show that, if you eat less sugar, you will not get cancer. or if you eat less sugar, your cancer will get treated. No, there is no information like that. And another very commonly asked question is cancer contagious? Like if somebody in my family has, will I get it? It's a big no. Okay, so cancer, full-blown cancer is because of multiple genes changes in an individual's body, which cannot be propagated to a person living along with you. Only those, even the virus spread, cancers caused by viruses do not normally spread. The virus causes genetic changes. Like I told you, it changes the proliferating genes. It also changes P53, but that cannot automatically happen if a person is living with it, unless again that same person is exposed to virus in a similar fashion. So cancer is not a contagious disease, and there is no scientific evidence that links attitude of a person to cancer. And does surgery cause spread of the cancer? Nearly no, because the resected area is generally just the fact of surgery. If it has already metastasized, then it would have gone to some other place. But surgery per se generally does not cause spread of this. There might be a very, very rare instance of mishap during the surgery, but that doesn't generally happen. Okay. And then these questions are asked, will cell phone cause cancer? Will electricity lights cause cancer? We do not have any scientific evidence towards that. We have to find out whether cell phone usage causes mutations in our body, which we do not still have evidence. And do herbal products help treat cancer? There is no scientific evidence yet about herbal products, plant products immediately treating your cancer. Okay, so I come to the end of my talk, and I would like to end by saying that the coming together of clinicians and scientists have helped us understand cancer better and come up with significant treatment modalities which have overcome a large number of cancers. But we still have miles to go before we conquer cancer fully, and we have a complete repertoire of answers to the title of my talk, why and how. So there's still a long way to go, and we have conquered only we can diagnose early and manage every form of cancer in humans. So I end by acknowledging and thanking the creative group, particularly to Supriya, for asking me to share this little bit of knowledge that I have with many of you, and I will look forward to helping in any other way to give talks which are related to life systems. I mean, cell biology particularly. Thank you one and all. I think we have time for a few questions.
Only few questions, ma'am. If any questions, if
They can
anybody
write to me.
wants to
Yes. They can write
interact
to
with
me also. Yes.
okay,
Unless there is a burning question, then immediate burning question.
you can ask question. Do it
I will
fast.
stop sharing.
Okay.
Yeah.
Actually, we
Is
are
there any
running out
question
of
in the
time.
box? In the box,
Uh,
yes. I think we lost a few minutes.
yes. Ma'am, before that, you tell me, ma'am, as you are a researcher, actually,
Yes.
where we are in this field, up to the point, or we have to reach more milestones regarding this topic.
See, the reason is there's no definite answer for that because cancer is not one single disease.
Mm.
Okay. Cancer is so many different disorders put together under one umbrella. So there are some types of cancers where we understand better, where we understand some bit of the genetics, For example, there is a cancer called a retinoblastoma. This is a cancer of the eye. It is a very severe, but we understand that because that is caused by one inherited genetic defect and another mutation that has formed. So there are some, similarly, breast cancer. Some genes have been associated with breast cancer. But whereas there are other cancers like pancreatic cancer and cancers of the liver, which are not caused by the hepatitis viruses, we don't know how to manage them so far. See, liver is a very remarkable tissue. There are very unique properties. I can give a whole talk on many of the slides that I have shown. Each one can make a 1 or 2 hours talk. So we have many new modalities. That's why I mentioned that, now it is no more a death sentence. Many of the cancers, if detected early, can be treated. But one thing we should remember is, even if 4 people have breast cancer, each one's breast cancer is different. One will be hormone responsive, one will not be hormone responsive. So that is the underlying difficulty of cancer management.
Okay. Ma'am, they have to be under monitorization, ma'am, after treating that cancer treatment. Means,
For initial phase, for each cancer, it differs. Uterus cancer, breast cancer, they generally recommend 1 or 2 years of continuous monitoring. Continuous means every 6 months or once in a year after the first 2 years. And then see, if you are event free for about 5 years, then you are as good as normal. So there are women who have had breast cancer treated when they were 30 years old and lived a normal life up to 70, 80 years old. So no more breast cancer is so much a cause for worry. So the last 20 years of management has now made cancer. See, the psychological impact has been very high, but that is not the case now so much. There is this pink groups which are popularizing early detection. And so early detection in the case of some of these cancers is very important, these solid tumors.
Okay. Ma'am, in case of one thing about the supplements they used to take, no, ma'am. After treating cancer treatment, they used to take some supplements, additional
What
supplements
kind
for
are they meant for general health improvement or any other supplement?
for a specific cancer treatment. They suggest some supplements regularly they have to take, no? For
No, what kind? I'm not aware. See, after
vitamins,
chemotherapy, your
proteins.
body becomes very weak because absorption from your intestine is very low. Okay. So there are some nutritional supplements that are given to quickly recover from the chemo effects. Okay, I am not aware of other supplements. See, because some kinds of cancer, for example, if you're talking about the therapy for blood cancers, where you actually get a transplant, then you are given supplements which actually inhibit the immune system because you have got a transplant. You have got bone marrow from some other individual. Okay, so then you have to repress the immune system so that it is not reacting to the cells that are given from outside. I don't know whether you mean that by supplements.
Yes, ma'am. That one.
Yeah, those are like, that is in special cases. Yes, you have to continuously take them. If you have had like kidney transplant, individuals also have to continuously take these immunoprotectants.
Yes, ma'am. Okay, I think it's already 4:30, ma'am.
Yeah. Yes.
We have to end the
Yeah.
session.
Yeah. Sure.
Dr. Radha, ma'am, your wealth of knowledge, passion for science, and dedication to unrevealing the mystery of cellular biology have enriched our understanding. We are truly privileged to have you share your expertise with us. We are very thankful for accepting our invitation and for being with us for this long time. Thank you. Thank you very much, ma'am. And thank you all the participants for joining with us. Okay. Shall I end, ma'am?
Thank you, ma'am.